Hello and welcome everyone. In this podcast we're going to talk specifically about mitosis and cytokinesis. I have learned over the years that mitosis and meiosis are two of the topics that students dread hearing about, and maybe it's because we talk about them in almost every biology class. But in genetics, particularly with meiosis, it's essential that you understand it. It's a really important concept. So even though you've had this before, let's go through this as if you haven't had it before, because you'll pick up some new things and it'll be important as we move forward for the rest of our discussions of the semester as we talk about mutations and then ultimately get into inheritance. Meiosis turns out to be really important. So you may ask, why are we talking about mitosis? Well, mitosis is a nice way to kind of jump into cell division. It's really important in understanding various kinds of mutations as well as cancer, which we'll talk about in a few podcasts down the road here. In the last podcast, we talked about the cell cycle. We talked about G1, talked about S, where the DNA is copied, G2 as it prepares for M phase, and we didn't spend a lot of time talking about M phase except to say that the chromosomes separate during M phase. But we're going to focus a lot on it in this podcast and the next podcast. These G1, G2, S, we might as well make this last statement about these two before we leave them alone for a while. Together we call these three cycles, parts of the cell cycle, interphase. And then we call this the M phase. So again, we're going to focus on M phase right now. And mitosis consists of 5 stages. The first one is prophase. The next one is metaphase. The third one is metaphase. The fourth is anaphase. And the last is telophase. Now I probably should have said 5 and maybe 6 stages. Some textbooks, I believe your textbooks, lump cytokinesis in with telophase. I'm going to list it separately. And the reason I like separating them out is that these phases up here deal with the chromosomes, what's happening with the chromosomes and to the nuclear membranes as well. Cytokinesis is the actual last part of the cell cycle where it physically separates out those two cytoplasms into 2 new cells. So I like to keep it separate. What I'd like to do is go through each of these phases looking at a pair of chromosomes as they go through. So let's erase this and draw some circles with chromosomes in them. Okay, so let's start with prophase. We probably won't get all of the stages on this one whiteboard, but we'll start. So prophase. And let's draw our chromosomes in here. And I want to draw 2 pairs of chromosomes. And I'm making one of the chromosomes, homologous chromosomes here, green and red. Remember, these are sister chromatids. The green and red are sister chromatids, and this green and red are sister chromatids. Over here, this is a separate chromosome. This is a blue and purple. Remember, these are sister chromatids, and this blue and this purple are sister chromatids. These are homologous chromosomes, and these are homologous chromosomes, just to remember those terms. So what happens during prophase? These chromatids condense. I should have drawn this circle just a little bit bigger, so I'm going to erase it and redraw it. And I want to draw the nuclear membrane around here. I'm going to draw it right now because at this point here, the nucleus, or I should say nuclear membrane here, begins to break down. Not completely, but partially. So let's just do this with it. Something else that happens are these centrosomes form. And they begin to form shorter kind of spindles from them. and they're going to begin to migrate to opposite ends of the pole of the cell. So centrosomes form. Now let's talk about pro-metaphase. Let's draw our big cell here again. One of the key things that's going to happen here is our centrosomes are going to have moved to opposite ends of the cell. And at this point the nuclear membrane has broken down, so that's no longer present, so we don't have to worry about that anymore. And so now we're going to have our chromatids here. And we'll have our other ones over here. And I'm purposely drawing them in not any particular pattern here. And as these centrosomes move to the polar side, so centrosomes at polar sides, opposite sides. Now, as these centrosomes have moved here, we often now call these the spindle poles, because they're going to shoot out, so to speak, these mitotic spindles, which I have written down here. And these mitotic spindles are going to attach to each of these centromeres. And anchor them. And this is a really important stage here. If this doesn't happen, the next stage can't happen and the following stages doesn't happen. It's very important to anchor each of these chromatids that are now attached to these mitotic spindles. Now remember, as we mentioned before, I'm not going to be able to draw them here, but these centromeres, what the spindles actually attached to, are going to be the kinetochore proteins that are always found at the centromeres. And that's probably enough for pro-metaphase. So let's go ahead and look at the next stage, which is metaphase. So of course we're still talking about mitosis and cytokinesis, but now we're going to talk about this third stage, that is metaphase. So let's draw our large cell here again, and we have metaphase. We have our spindle poles still at opposite sides. For our purpose, we're just going to continue to call these spindle poles. On the last whiteboard, we saw that the mitotic spindles, which are actually, remember, microtubules, attach to those different chromatids. And now what's going to happen in metaphase is we're going to begin to align these chromosomes down the metaphase plate. And I'm just going to draw a dot line. There's no real physical appearance of what this metaphase plate is, but I think we should write it down here. And in doing so, what happens is each of the sister chromatids are going to end up on either side of the plate, of this metaphase plate. So let's go ahead and draw them like so. Now, important to remember, it's kind of hard to draw this exactly the way I want to, but these still are attached at the centromeres. So they're still attached right here at the centromeres. It's kind of hard to draw exactly how it appears, but they are still attached. Then we have our smaller ones here, and remember these are still attached, just like the larger chromosomes up there. It's just hard to show it exactly the way it's supposed to be. Okay, so the key thing here is these spindles have positioned these chromosomes such that on each side of the metaphase plate there is a sister chromatid. Your book talks about different names for these spindles at different stages and what they're doing, but for our purposes we can just call these mitotic spindles. Before we move to anaphase, I'm going to reposition this term here, metaphase plate, into a description here of what's happening here. I drew the picture, but let's go ahead and just write it out in words next. What's happening during metaphase is that the spindles have positioned the chromosomes, abbreviated, so that each sister chromatid, I'm going to abbreviate that SC, so that each sister chromatid is on opposite sides of the metaphase plate. So what happens next is anaphase. And in anaphase, our spindle poles, here and here, begin to pull apart the chromosomes. They begin to separate these two sister chromatids from each other. I'm going to go ahead and draw the metaphase plate again, just for a landmark for us. But these spindles begin to shrink. And remember, over here, they're attached to each of these sister chromatids. And as they shrink, they pull those sister chromatids apart from each other, such that you have the one here, and the shorter chromosomes here. So we can say what's happening here. These mitotic spindles shrink. And what that does is it separates the sister chromatids and pulls each to opposite poles. Okay, now we're ready to talk about telophase and then cytokinesis. Alright, still talking about mitosis and cytokinesis, but now we've moved almost to the end game here. So we're going to draw our cell again here, and this time I'm going to draw the cells a little different, because what we're going to see here during telophase is that the chromosomes have now each made it to their destination here. The nuclear membrane now begins to reform around each one, as shown here. So chromosomes reach their destination. The nucleus reforms, and the chromosomes now decondense. And then to finish things off, let's talk about cytokinesis just real quick here. And essentially, we finish separating these two cytoplasms apart with two new cells here, each with their respective nuclei and their chromosomes in them, and now they can re-enter the cell cycle and divide again. The end result are two identical cells, genetically speaking, as their original parent cell. Now what I'd like to do is go back through these and talk about the number of chromosomes and chromatids that are present at each stage. So chromosomes, chromatids. Okay, so in this cell we have 1, 2, 3, 4 in prophase. So we have 4 chromosomes. Number of chromatids, we have 1, 2, 3, 4, 5, 6, 7, 8. We have 8. Over here we have the same situation. The chromosomes are still together, and as long as they're together, we're going to call them 4 chromosomes. And we'll still have 8 chromatids. As long as the chromosomes are held together at the centromere, they will be counted as the same chromosome. So let's do the same here. So in metaphase, again, we have 1, 2, 3, 4 chromosomes. Remember, they look like they're separated, but it's just the way I drew them. There's still 4 separate chromosomes here. And we have 8 chromatids. Now at anaphase, they begin to be pulled apart, and it's still the same cell. So at anaphase, we now have 1, 2, 3, 4, 5, 6, 7, 8. We now have 8 chromosomes. And we also have the same number of chromatids, 8. So now let's look at telophase. So at telophase, so this is kind of a tricky one here with telophase, because they have now formed in their own nuclei, but it's still the same cell because the cytoplasm has not formed. So it depends on how I word the question. I'm more likely to word the question in a way that says, how many chromosomes or chromatids are in a cell at telophase? And if I say a cell, then you consider this all one cell. So in this case, we're going to have 8 chromosomes. And we'll have 8 chromatids. Now, after the cell is divided and we're in cytokinesis, you would say we have 4 chromosomes per cell and 4 chromatids per cell. So 4 in here and 4 in here. Okay, before we move on to the end of this podcast, I think it's important to talk about the outcomes of mitosis. We've said some of these already in passing, but let's go and just make a quick list here. One is that you produce 2 identical cells. Genetically identical cells. It's possible that, particularly during embryonic development, when you go through mitosis, that the cells might end up with different functions, but genetically they're the same. New cells have the same chromosome complement. So you go from a diploid to a diploid. It's also important to remember that as the cell's dividing, it's not all about the nucleus and the chromosomes. There's other things there that also need to make sure they make it into the next cell. So the new cells each have a complement of cytoplasm. So as those new cells are formed, they're going to have each have to have mitochondria, Golgi, ER, and all the other cytoplasmic components and organelles present in each in a sufficient quantity so that cell can continue to do its job. If a cell divides and it only ends up with half the mitochondria, as it's supposed to have, that's going to have a consequence to the new cells. When does mitosis occur? Just kind of as a quick reminder and summary here. Mitosis occurs, in fact it's incredibly important, right? Because you have your one-celled zygote here, fertilized egg, and it will grow and continue to grow and continue to grow until you have a child and then ultimately an adult, right? So you go from one cell to trillions of cells, and this is all because of our good, reliable friend mitosis. As these cells divide, they continue to make new cells that are genetic clones. No genetic diversity. They may express different genes, which allows the cells that begin to specialize in this human to have a different function, but genetically they're identical as the zygote. Now in a few days we're going to talk about mutations, and sometimes mutations can make it that so they're not genetic clones, but for now we can just say that this is always going to produce a genetic clone. It's also really important, as this individual here, to place dead cells. So as a fully formed organism lives and breathes, their cells will continuously need to be replaced. Either they die because they have reached the end of their particular cellular life, or they're damaged because there's some kind of trauma that occurs. When that happens, these cells need to be replaced, and they're replaced by using mitosis. Alright, so what should we have learned in this podcast? You should know the 5 stages of mitosis, plus cytokinesis. With each of these stages, you should know, so with each stage, know what's happening with the chromosome as it's moving from one stage to the next stage. You should be able to count the number of chromosomes and the number of chromatids at each stage. You should also know the outcome of mitosis. And you should also know which cells, sort of the importance of mitosis, right? So which cells undergo mitosis. Alright, that's all I have for this podcast. If you have any questions at all, please make sure you come and see me. I'll see you in class.